The natural killer cells are actually your first line of defense against viruses, bacteria, tumor cells that are in your body, swimming around your body. And what happens is the natural killer cells are the first ones who come and who attack, who see that this cell is foreign in the body, and they start attacking. And then what happens is they end up calling in the adaptive immune, so that's called the innate immune system. Then as they attack these foreign cells, they also call in the adaptive immune system, so the T cells, etc., to come and help them kill foreign cells. So that's natural killer cells. And what's important here is that there's a body of data, decades old, to show that natural killer cells are involved or are decreased in a number of cancers. So there's a large body of evidence to show that natural killer cell activity is low in a number of different disease states, such as cancer. Because we knew that natural killer cell activity, based on the published literature, was low in cancer, we wanted to see for our test, which was developed, by the way, in our global company in Korea. What they had shown was that with our test, we were able to replicate what was in the published literature, that there was a significant difference between a control population and people with different kinds of cancer. Now, the tests used in research for detecting natural killer cell activity are very complicated. So there's the gold standard, which is the chromium-51 assay. Now that is, you have to isolate cells, you have to incubate them with a tumor cell line that's been preloaded with radioactivity, and then you measure radioactivity. So that's a test that's only really used in research. There's no real commercial application. Our test is a very simple blood test. So basically you take 1 ml of blood, which has in the special tube, there is a cytokine cocktail that's been patented to be stable, and then you take that with the blood, you incubate it for 20 to 24 hours at 37 degrees, and that allows the activation of the natural killer cells to start making interferon gamma. At the end, you take the plasma and you measure IFN gamma in an ELISA, a simple ELISA. So it's a very simple test. So we replicated in different cancers in Korea, that we were able to see that difference between a control population and a cancer patient. So then we said, okay, so we wanted to then look in a North American population. Let's do a similar study looking at patients going for colorectal cancer, for colonoscopies, basically. So the first study that we did was to look at patients coming into a hospital called Hôpital Maisonneuve-Rosemont in Montreal, and it's patients scheduled for colonoscopy. And these were patients, they weren't an average risk population. It was kind of a bit of a higher risk population, people with either a positive fecal test, rectal bleeding, anemia, and some of them were screening, but by and large it was a sort of moderate to higher risk population. And we screened we tested over 1,000 subjects coming in for their colonoscopy, but each one had an NK view, so the natural killer cell activity test, which is called NK view, before their colonoscopy. So the primary result is that we were able to show that our test was highly sensitive for the detection of colorectal cancer. So we showed that we had 85.7% sensitivity for CRC. What that means is somebody who has colorectal cancer, if you do the test on them, 85.7% of the time you will be able to detect that CRC, or that cancer, colorectal cancer. For the moment, it's approved in Canada, and it is available in Canada. In the US, it's available as a lab-directed diagnostic in a lab in California. So in Canada, it is freely available. So a physician could order the test. It's something that's simple. You could have it done at the doctor's office, and then after the incubation, you take that plasma, and the lab comes and picks it up and does the ELISA assay. In Canada, we have, in different areas, some issues with waiting lists for people who are waiting for colonoscopies, and they're waiting a little bit longer. So in this way, the physician can maybe prioritize who should go for a colonoscopy first. Not that they're not going to go for a colonoscopy, they will. It's just a question of prioritizing. Because the other thing that we showed in this study is that it had a very high negative predictive value. What that means is if somebody is negative on the test, there's a very, very high likelihood that they won't have a cancer. In other words, that they will be healthy. So that is one way that you can sort of use the data from out of this trial to decide when to prescribe the test. This is another way, or a very different way, of looking at a risk assessment of somebody for colorectal cancer. It measures something different than the fecal test. The fecal test measures blood and stool. We measure the activity of your innate immune system, which is very different. So it's another tool in your armament of what you have available to decide what is the risk that my patient might have colorectal cancer.